Hi everyone! In this video, we'll look at the stages of meiosis and in particular the behaviour and appearance of the chromosomes during each phase. It may be worth looking over the phases of mitosis before exploring this content. You should also ensure that you're familiar with the concept of homologous chromosomes, which was covered in our last video. There are two divisions within meiosis, and we refer to these as meiosis 1 and meiosis 2. The names of the stages are the same as those we've seen in mitosis, but each occurs twice. The first stage of meiosis is therefore prophase 1, when the chromosomes condense and become visible, and homologous chromosomes pair up to form bivalents. During this phase, the nuclear membrane also breaks down and the spindle begins to form. The next stage is metaphase 1. As in mitosis, the chromosomes line up at the equator of the cell. However, within meiosis 1, the chromosomes are lined up in their homologous pairs. The spindle fibers attach to the chromosomes at their centromere. Next, we enter anaphase 1. The spindle fibers contract, pulling each of the chromosomes within a pair to opposite poles of the cell. The final stage of the first division is telophase 1. The chromosomes have arrived at the poles and decondense. The spindle disappears, and this is followed by the splitting of the cytoplasm in the process of cytokinesis. This completes the first division. Meiosis 2 begins with prophase 2. Once again, the chromosomes condense and become visible, and the nuclear membrane breaks down. During metaphase 2, the chromosomes line up at the equator, but here they lined up in single file as we see in mitosis, as there are no longer homologous pairs within the cell. The spindle attaches to the centromere of each chromosome. During anaphase 2, the spindle contracts, this time splitting each chromosome at the centromere and pulling each chromatid to opposite poles of the cell. The final stage is telophase 2. The chromosomes arrive at the poles and decondense. Cytokinesis once again occurs. This type of division results in 4 haploid daughter cells, and therefore each having only one copy of each gene. In the next couple of videos, we'll look at the events of meiosis in more detail to reveal how further genetic diversity can be introduced. The first of these will examine the process of crossing over.